Good morning and good afternoon, everyone. Welcome to this workshop organized by Metabolomics Workbench in, in coordination with our CFD-related work. Today we are going to talk about the work that we have been carrying out over the past 20 years, and more recently, its integration with the NIS recently developed center called Common Fund Data Ecosystem. So this work really has been more than 20 years of a journey, where it started with this NIH program called Lipid Maps, which lasted for about almost 6-7 years, followed by Metabolomics Workbench, our national metabolomics data repository. And then more recently, we have joined the effort with another NIH program called Common Fund Data Ecosystem, whose objective is to integrate, provide a platform for integrating data and knowledge from many such data resource centers, such as the Metabolomics Workbench. So why we need to study metabolomics? Metabolites are essentially a functional endpoint of physiology and pathophysiology. For example, starting with the mRNA transcription, then RNA translation into amino acids, followed by proteins, their activation. Some of those proteins act as a metabolic enzyme, and they convert different metabolites into other forms. Good thing about metabolites is that, relatively as compared to many other types of molecules, they can be easily measured from serum. And they, in some sense, they also are a direct mirror of environmental influences, such as what we eat, how we exercise, and what medicines we take. So the end objective is that can we metabotype quantitatively all the individuals so that we can monitor their health and disease. So what are the challenges in metabolomics? How quantitative is the field of metabolomics? How can we analyze the omics measurements to obtain metabolite information? How can we organize, store, and use metabolomics data? And then how can metabolomics data be integrated with other omics data to provide a systems view of the biology? More importantly, what are its implications for systems medicine? Can we metabotype an individual? To address all these questions, almost 13 years ago, NIH started the Common Fund Metabolomics program to address issues such as training in metabolomics, metabolomics technology development, comprehensive metabolomics resources, metabolomics reference standards and synthesis, metabolomics data sharing, and international collaboration. So it resulted in Metabolomics Workbench, our national metabolomics data repository, which our group manages. What I have shown here is a screenshot of the Metabolomics Workbench MMDR portal, which Dr. Oin Fahe will talk in a lot more detail pretty soon. Now, NIH, after starting several such centers related to metabolomics, proteomics, gene expression measurements, and so on, then they realized that there is a need to be able to integrate data from all these different types of data, and which collect data in many different modalities, to derive biological knowledge. And with that objective, several years ago, they started what is known as NIH Common Fund Data Ecosystem, of which we are also a part of. More recently, in the last year, CFD entered phase 2, where they started organizing the whole CFD into a couple of dedicated centers, such as Data Resource Center, and soon we will have Cloud Implementation Center, Center for Training, Knowledge Center, and Integration and Coordination Center. We have the portal for CFDE, the link for the portal for the CFDE is shown here. It's https://info.cfde.cloud, where everyone can find more information. Within CFDE, we are also trying to integrate data with many other resources, such as all of us, TEDI, dbGAP, etc. So in this workshop, we are going to cover many aspects of the work that we do at Metabolomics Workbench as a data coordinating center, as well as our interaction with the and participation in the common funded data ecosystem. So without further delay, here is the detailed schedule for the workshop, and I transfer the control to Dr. Wayne Fay, where he will talk more about Metabolomics Workbench.
All right, good morning, good afternoon, everyone. I'm going to give an overview of metabolomics in general before we go into the details of the workbench and the NMDR. So as Mano mentioned, this project got underway about 11 years ago, 2012, 2013, and it's centered at UC San Diego. We had a number of other collaborators around the U.S., and over the years, it's grown to an international consortium. And before I start with the science, I just want to go over some definitions. So metabolomics is basically the concern with the high throughput identification of small molecules in the metabolome, as opposed to studying individual structural molecules like natural products, elucidation, and things like that. So really, we're talking about high throughput identification of many metabolites at the same time. So the other buzzword is metabolome. So that can be applied to either a tissue, organ, a cell, or typically a species too. So it can have many different types of metabolomes, and those are all closely related to the genome, the transcriptome, and the proteome. So the metabolites themselves are typically small molecules under 1,500 Daltons in size that are substrates or products or intermediates of metabolism. This 1,500 Daltons, it's not a hard number, but it correlates very well with the range, the mass range of mass spectrometers. So it's a convenient range to study in terms of metabolomics. So here are the main building blocks of biology. You have the genome transcribed to the RNA, translated to the proteins, many of which are enzymes, which in turn give rise to various metabolites, sugars, nucleotides, amino acids, lipids, and many others. But these are the drivers mainly of phenotype and function, which is why they're so interesting, because the metabolites are where the rubber meets the road in many diseases and conditions that we're interested in looking at. A couple of more definitions. A primary metabolite are those associated with normal growth, development, and reproduction, especially in mammalian systems, but also in plants too. So examples are cholesterol, glucose, tyrosine, and so on. So the major metabolomic pathways. In contrast, a secondary metabolite is a molecule that's not directly involved in these key processes, but may have other functions like they may act as defense chemicals, pheromones, pigments. They're commonly found in plants, microorganisms. So typically, most of metabolomics is devoted to the study of primary metabolites, but not all. A couple more definitions that you frequently encounter are targeted metabolomics versus untargeted. So in targeted metabolomics, one is specifically looking at a set of metabolites for which they usually have standards, synthetic standards, and they would run concentration curves. It'd have exact information in terms of retention time, LC retention time, MS fragmentation, and so on, in order to get quantitative measurements. Whereas untargeted metabolomics is the concept where you're trying to detect as many metabolites as possible, whether or not you have standards for them. So these typically are semi-quantitative measurements like relative peak area, peak height, and obviously typical untargeted metabolomics study would have many, many more identifications. So the scope of metabolomics is really important. There are the endogenous metabolites existing in that organism. These are the ones that are synthesized by that organism. There are the metabolites that come from the diet. There are drugs in case of mammals, prescribed or not. And then we have the other commensal organisms like the microbiome from bacteria, fungi, and other microbial organisms. And last but not least, metabolites in the air, water, that's considered the exposure. So really, when you're measuring, say, a blood sample, you can, in principle, measure all of these components. In terms of the major taxonomic categories in the studies that we've received at the NNDR, it's very vast, but two really stand out. Obviously, human and mouse. Mouse is the major mammalian research organism. So these two take up a big chunk of the pie, but there are others too: bacteria, fungi, plants, fish, insects, and others. In terms of the sample source, this is another important area because the metabolome of each sample source is going to be quite different. Blood is the most commonly encountered sample source, followed by liver, culture cells, urine, feces, and so on. And in terms of disease association, of all the studies we've received, cancer is by far the largest. There are over 200 different disease associations that have been studied that we have data for. So it's very, very comprehensive. So I want to talk a little bit about classification of metabolites because we're going to deal with this in the later talks. There's no official classification system for all metabolites. Now there is for lipids, which comprise a large component of the metabolome. This was formulated by lipid maps over the last 21 years and is very well internationally accepted. So you can see the top 7 categories here are all lipids. As well as that, we have nucleic acids, carbohydrates, sugars, alkaloids, benzenoids, organic acids, and others. So let's look at the distribution of metabolite classes. So lipids comprise of almost 2/3 of the detected metabolite classes in metabolomic studies. This is largely driven by the fact that lipid chains are highly diverse, chain length, double bonds, so on and so forth. So this really drives the numbers up. Also, another huge category are the organic acids, including amino acids, peptides, nucleic acids, and so on. So how do we classify metabolites? Well, like I mentioned, we have the lipid maps classification to handle the lipids. For the non-lipids, we use a software called Classifier developed at the University of Alberta. These are partially successful, and we follow that up with the curation of these uncurated classes. So, for example, we've created new super classes for nucleic acids, carbohydrates, and so on and so forth. So all of these go into a nomenclature database called RefMet that I'll deal with in the next talk. So these are some examples of the super classes. These are the lipids. As you can see, they're drawn in a very consistent fashion. The business end, like the carboxyl group or the head group, is on the right-hand side, and the chains are on the left. So this is very important, for example, for sphingomyelin and phosphatidylcholine. These are two entirely different molecules with different biosynthetic pathways, but when you represent them in a similar fashion, you can quickly see the similarities, the choline head group, the chains, and so on. Another lipid superclass are the polyketides. They're very often found in plants, microorganisms, and so on. For the nucleic acids, obviously, purine and pyrimidine are hugely important in terms of the nucleotide bases. adenine, guanine for the purines, cytosine, thymidine, and uracil for the pyrimidines. And we have other coenzymes such as FAD and NAD that do contain a nucleic acid component on the right-hand side. Organic acids are another big category because they comprise amino acids and peptides, dipeptides, tripeptides. Also the TCA acids and in the citric acid cycle, keto acids, and so on and so forth. Carbohydrates, another very big class. We deal with the short carbohydrates, either monosaccharides, or trisaccharides, and obviously not the oligomers, because they would exceed the mass range. And for alkaloids, these are nitrogen-containing molecules that have a ring. So you have examples are benzodiazepine, niacinamide. And then benzenoids are also commonly found, and a number of others that I won't go into. But we have a metabolite classification system on the Metabolomics Workbench where one can look at all of these classes that I just mentioned. You can search them, look at the core structures, and so on. So what does a metabolomics study workflow look like? Well, you start with study design, which is very important in terms of what exactly the researcher is trying to do. There are various treatment sample prep stages. So and typically there's two main types of instrumentation, mass spectrometry and NMR, which then goes on to data processing and data analysis. So looking at the processing side, we have data pre-processing, where you do peak picking, deconvolution, etc. Analysis of that, chemical property prediction, metabolite identification, either with vendor software or custom workflows. And then you go into the details of analyzing that data in terms of the functional analysis and metabolic modeling. So every institute has a different workflow. Well, most of them do have different workflows. So there's very large amounts of software, especially in the last 10 years, that have been developed to do this post-processing step. What are the main types of instrumentation? Mass spec by far is the most common. So it's about 90%. And of that 90%, LC-MS is definitely the most frequently used system, either with reverse phase chromatography or helic chromatography. So what are people trying to look at? Well, some people have a very broad approach. They're trying to study all possible pathways, say in human blood or human liver. And obviously the scope is very, very large. Here's a large scale metabolic map of the types of molecules one could look at. A little bit more focused is lipid metabolism. This is a study that was done by the Lipid Maps Consortium a number of years ago, where certain major classes of lipids were studied in macrophages. So this is still very large scale. The numbers here refer in orange refer to the numbers of these lipid classes that were detected and quantitated. And obviously there's interplay between all these pathways. And then the third and more focused approach would be, say, one particular pathway, in this case, the arachidonic acid cascade. So this would be an example of a targeted assay where the researcher would have synthetic standards for all of these prostaglandins and heats, and one could get quantitative measurements in terms of micromole per gram, micromole per volume, whatever. So in terms of metabolomics and disease, we have on the workbench, we have a lot of studies that are directly studying the interplay between metabolism and disease. So for example, people look at cancer. That's probably the most commonly studied disease. They may be looking at the Warburg effect, where you have the citric, glycolysis is upregulated, and one can monitor that with all of the glycolytic metabolites and also the metabolites involved in the Krebs cycle in the mitochondria. Another very interesting area are inborn errors of metabolism. So in the case of sphingolipids, various enzymes involved in the degradation of glycosphingolipids are modified. So for all of these diseases in the yellow boxes, one particular enzyme in the cascade of degradation of sphingolipids is interrupted. So and ending up in a buildup of usually of the substrate and less of the product. So one can monitor many of these diseases by looking at the glycosphingolipid profile. Another example of a inborn error of metabolism is phenylketonuria. So this is a well-known one where phenylalanine hydroxylase cannot convert phenylalanine to tyrosine with buildup of phenylalanine. So one can measure these types of processes easily by metabolomics and spectrometry. And diabetes is another huge area of interest. And again, some major glycolytic and TCA cycle pathways are affected. So typically in diabetes and obesity studies, the researchers are homing in on these major pathways occurring in the cytoplasm and the mitochondria. And one other one is, which is very relevant right now, is cholesterol and bile acid synthesis, where you have an interplay with the gut microorganisms, the bacteria, which can, for example, convert various bile acids to secondary bile acids. And so you have an interplay between the host metabolic processes and the bacterial metabolic processes. So that concludes this portion of the talk. If there are any questions, I can go on to the next part.
If attendees want to ask a question by speaking, please raise your hand, then I will unmute you. Or you can type the question in the Q&A panel, and then we will try to answer. You can also send messages via the chat channel to the hosts and panelists.
On to the main topic here is the discussion of the NMDR, the National Metabolomics Data Repository, which was established in 2013, about 11 years ago, as a repository for the Metabolomics Workbench. So the main portal is the Workbench website. So the home page always gives the latest number of studies. It's probably different today than it was 3 days ago when I made this slide. But that top panel shows the total number of studies, the number of publicly available studies, and some examples of the latest release studies. So here you can see that the growth of NMDR over this 11-year period. We started in 2013. It was limited to a number of researchers in the US who were funded by the Metabolomics Workbench grant. But then in 2014, 2015, it was opened up to the rest of the world. We introduced an online deposition system. So now it's taken off to the point that we have over 3,000 studies right now. Looking at some of the numbers here, this is a few days ago, you can see the total number of studies, 3,248, numbers of samples, 338,000. Total number of named metabolites submitted is almost 700,000. The map to the nomenclature system, which I'll talk about later, is this number. And the numbers of different species is over 300. So a lot of different species. Like I mentioned, human and mouse are the main ones. numbers of disease associations, and numbers of sample sources. We have about 450,000 raw files, comprising about 50 terabytes of raw data, which is very important in terms of being available for reuse and reanalysis. How do we set up the whole system? We have a public website that people can access. We have a data submission portal. So when submitters send us data, they have limited access to their studies until they're released. And then on the right-hand side, we have development websites where we curate and do various things in the background. So an overview of the inputs and outputs. So we have the online data submission system up on the left, where metadata, targeted measurements, methods, files, and so on are deposited via the web. We get the raw data also from the submitters via FTP, and the metadata and name metabolites are entered into a relational database, where on the output side, then we have various web pages for browsing, searching, statistical analysis. We'll go into detail about that. You can download targeted and untargeted measurements as text files. You can download the raw data, very importantly. You can download what's known as an MW-tab file, which I'll talk about a bit more, which basically encapsulates all of the components of a study. And then we have a REST service, where one can computationally interact with the NMDR. So a typical workflow for metabolomics is you have a design. You design your experiments, which describe the sample origin characteristics. You describe the treatments, collection, sample prep, instrument analysis, and data pre-processing. So all of this has to be captured by the NMDR to be able to describe a study and hopefully allow one to repeat the study and at least understand in detail what the samples have been put through. So here's another overview of the strategy. Up on the top, you have the data coming in through the online submission system. For name metabolite data, that goes directly into a relation database. For untargeted data, these are large data sets, maybe 10, 15, 20,000 identifications, M/Z retention time pairs. These are kept as files. The metadata is a database, but the files are then put in a big data file system, further processed, and then analyzed in much the same way as for the name metabolite data. So whether name metabolites or untargeted data are submitted, many of the data analysis tools are the same. So stepwise, what this involves is a submitter adds data for the project, the study, the subject, the study design, what the samples are, control, disease, treatment, etc. Collection, treatment, sample prep. Then there's a decision point here. Is it NMR data or is it mass spec data? If it's mass spec data, what are the chromatography methods? What are how many mass spec methods and what type? The product of these are the number of analyses, and each analysis then needs its own set of results, which are then entered. All of these are entered into what we call an MW-tab file, which is a large text file. So this is really the common currency behind the scenes for metadata, data sharing, and storage. It contains all these sections on the left that the submitter has just entered, as well as names, metabolite measurements, and annotations. Now, it would not contain untargeted data like M/Z retention time pairs. They will be in separate files, and it obviously does not contain the raw data. But it's a format that's human readable. It's similar to the UniProt format for proteins, and it can be easily parsed by computer programs. and download it. So, and it has its own MWTab file format specification. So someone could actually create an MWTab file with data and submit that to the workbench if they knew exactly what the specification was and are careful about all the crossing all the I's and dotting all the T's. Okay, so for an online submission, normally one will create an account, register, log in, do a new data upload, enter the relevant information in a form, and then start the submission. If it's a brand new submission, they'll start an online submission here. If it's a Metabolon submission, then they have Metabolon has a special template. Importantly, if you've already submitted a study before and the parameters are very much the same, then you could use that previous study as a template. So you only have to change the things that are different. You know, obviously the study summary, the study design, and other parameters, and everything else then is the same. I'm not going to go through all of this, but this is an example of one particular form for the project, and all the pink items are required fields. So you have to enter all of these, and then the other ones are optional. Obviously, the more information, the better. All the tabs up on the top indicate all the steps that you have to go through to finalize the study. And when all of that is complete, and when the study is curated and placed on the workbench, then you can access all of the data through the browse and search studies from the data repository tab. So if you're on the website right now, go to data repository, browse, search, and studies, and summary of all studies shows all of the currently available studies. The other way to do it is to look at projects. So studies with the same theme can be grouped into a project. So for example, if somebody is doing some cancer study on mice, and they want to separate their data into the liver study versus the blood study, or whatever criteria they want to break it into, but it belongs to the same project, then we can designate all of those studies within one project. So then you can easily click on that project to see all the studies belonging to that. So that panel in the bottom left here shows this project 1288 has many studies associated with it. So the NNBR study level view also contains multiple metadata sections. So just like those that were entered. So all of these tabs can bring you to those particular portions of the metadata. And at the study level view, if you want to look at name metabolites for a name metabolite study, you click on this link. It'll give you the list of metabolites. Then you can get into more detail by selecting one metabolite of interest, let's say cholesterol ester 16:0, and see all the actual measurements for that sample. Look at the ANOVA data for that sample across, in this case, treatments. Look at the t-tests. Look at a box and whisker plot for all the different parameters that are changed here, the different, what we call factors, and look at the data that way, and also look at an entire box and whisker plot. So those are all from the main study page. Importantly, you can download data. You can see the samples. You can download the metabolite data as a table. So if you want to do your own analysis on it, you can download the MWTab file that I just talked about that contains not only the results, the named results, but all the metadata too, in either plain text or JSON. So you have a lot of options there. Study-specific tools, should go back one, perform statistical analysis. I will go into more detail later on, not in this talk, about all the study-specific tools and general metabolomics tools. Raw data is really important. We have a link to raw data for every study. So most of the raw data we have is LCMS data, but also some NMR. We have something like 50 terabytes of raw data available. Submitters are encouraged to upload raw data in open source, because then researchers can use it much more conveniently without needing to have the vendor-specific software. And one important aspect of raw data availability is the ability to reanalyze raw data when new standards or validated mass spectral data become available in the future. And that's definitely happened in the last few years. So if you go to the raw data link for a study, you can view the contents. So this saves you the trouble of having to download the entire raw file, which could be very large. It could be gigabytes in size, and you're able to see what's in the file. These in the sample table on the workbench, there's a link between the individual sample and the raw file, so that will tell you, that will guide you as to what each raw file refers to in terms of samples. So you can download an individual raw file. You can also view that raw file with, if it's open source, and actually look at the MS1, MS2 features, look at the TIC plot, look at the MS2 plot, look, and actually search for product ions or precursor ions. Even better, you can run it in GNPS. GNPS is an initiative also at UC San Diego, Global Natural Products Center, where they have much more sophisticated tools for looking at tandem mass spec data in terms of graphic output and networking. And another option is to run do a search of MS2 spectra in ADAP-KDB. This is Zhu Du's group at UNC Charlotte, where they've developed software in collaboration with the Metabolomics Workbench to do various things like search the raw spectra against public libraries and group raw data in terms of clustering and developing a knowledge base. So that's another very interesting thing that you can do on your own with individual raw data files. In terms of searching data.
Hey, Ryan, let me interrupt you here. So it has been about 15 minutes, so perhaps we should just pause for a minute or two to ask any questions.
Okay, cool.
There is one question that Dr. Ashok Gelhot asked, and the question is, do you have work on sulfur metabolism biosynthetic pathways?
Do we say that again, Mano?
Do we have work on sulfur metabolism biosynthetic pathways? Which I suppose means if in our pathway database that we have, is there a pathway related to sulfur metabolism? So
what's that first word before metabolism?
Sulfur metabolism.
Sulfur.
Yeah.
Not that I know of. We have we have links to major pathways, human pathways through KEGG, but off the top of my head, I don't know about sulfur. The interest is mainly driven by mammalian pathways, I think. So I think sulfur metabolism, I'm not sure exactly what metabolites he's referring to, but off the top of my head, I wouldn't know.
Okay. If there are any other questions, please post them in the Q&A channel. That is the preferred mode, although you can also post in the chat. So we will wait for maybe another 1 minute for any additional questions. I think you can proceed, Owen, and maybe in about another 15 minutes, we can take just a 2 minutes break for any questions audience might have.
Sounds good. So what we're looking at here is the quick search bar. You'll see that in the top right portion of the Metabolomics Workbench website. And this is by far the easiest way to look for anything, especially if it's a metabolite name or a study ID. So by far the easiest way to access a study ID is just to type in, ST00001, whatever it is. Metabolite name, PubChem compound ID, that's a number if it's in our database. Metabolite mass, so that's in a range of 50 to 1500. It'll match plus or minus 0.5 Daltons by default. Metabolite class, anything in a study title. So any word like cancer, obesity, any word that would be found in a study title, you can search for that way. And it will, if it's a molecule, if it's metabolite, it'll give an output. You can see down the right, the RefMet name, that's the standardized name. The metabolite matches for that particular name in a database. So there's 18 different matches to citric acid in our database. So citric acid, isocitric acid, and so on. And then importantly, all of these matches to citric acid in the study. So all of these, in this case, there are 661 studies that have detected citric acid. So that's important too. For those who have the necessary skills, you can do a regular expression search by prefixing grep : and doing a grep search. So for this particular example, any words that start with 2-oxo and end with acid are picked up here by this regular expression search. Very easy way to access the studies is through the bubble chart access. So that's from that main panel for the browsing studies. So you can search by species, disease, sample source, and metabolite class. You just click on a bubble and get the data. So for example, for sample source, you can either click on a bubble, say for urine, or in the table below, click on the urine value. So that gives you the list of all of the studies that contain urine as a sample source. So it's a very fast way of getting that type of data, rather than having to type it in and do a search. You can text search on NMDR studies. For this example here, you can search for all studies containing NAFLD, non-alcoholic fatty liver disease, in the title. And you can then filter the search to say human, an institution, year submitted, so on and so forth, to get a much shorter list. You can do a power search on all experimental metadata fields. It could be in a collection field, it could be a treatment field, anything like that. So the example here is NAFLD. And down at the bottom, there's a list of studies where the word NAFLD has appeared, sometimes in the study title, sometimes in the treatment study, so on and so forth. So it's a kind of an in-depth way of searching for one particular word or partial word. I want to switch now and talk about standardized nomenclature. We have a system called RefMet, which is a standardized nomenclature, and this is a huge problem in metabolomics. Unlike genomics and proteomics, where you have a very controlled way of defining genes and proteins using well-known and well-accepted identifiers like the Entrez system. In metabolomics, it's much more like the Wild West. Everybody has their own way of naming metabolites and their own workflows. So it's a huge task to be able to harmonize all of these to discern which metabolites have been detected similarly across studies. So what we've done with our over 3,000 studies is to develop a very highly curated analytical chemistry-centric database. By that, I mean all the names in our nomenclature system are coming from real studies as opposed from just downloading metabolite names from big databases like PubChem, HMDB, KEBI, and so on and so forth. So that's a very important feature of this RefMed. It's based on real data. So what are the sources of metabolite name heterogeneity? You have synonyms. Everybody has these: alpha-ketoglutarate, AKG, and so on and so forth. You have two-part name submissions, adenosine triphosphate, ATP. We get suffixes with the anion adducts, all the MS adducts attached to the end. We have isotope suffixes. We have user-specific information, so they detect this metabolite TG54:2 at 2 different retention times, for example, and submitters will label it like this. We have other suffixes containing retention time information. There's many different types of proprietary mass spec processing software, different open source software. All of these contribute to huge metabolite name heterogeneity. And so this is an example of all the synonyms we get for just one RefMed name, LPC 16:0. So as you can tell, if submitters are giving us all of these different synonyms, it's a real disaster in terms of being able to compare and contrast unless you have a good nomenclature system. Here are more examples of the types of names we get coming in the door, and so the job of RefMed is to harmonize all of these to one easily recognizable name that most people can understand very quickly. This is a very important slide in terms of the annotation levels from a metabolomics experiment. So for targeted experiments, typically people can actually discern the exact structure, double bond positions, and so on, and stereochemistry using standards. And the next level is the regiochemistry level, where, for example, the chirality might not be known. But again, one can draw a structure for these. Then you get down to untargeted experiments where you have information on the molecular species level, not the exact structure. So for example, for a phosphatidylcholine, the researcher may know that it contains 16:0 and 18:1 fatty acyl chains, but not the SN regiochemistry, and not the positions of double bonds or the geometry of double bonds. Other examples are citric acid, isocitric acid, leucine, isoleucine. So no exact structures can be generated from these. Then the lowest level is one can determine the metabolite class, whether it's a phosphatidylcholine, ceramide, and so on and so forth, and tell the total number of carbons and double bonds, but no further information. So this is the other problem we're faced with, different levels of metabolite certainty and confidence. And metabolomics has to be able to handle all 4 of these and have a nomenclature system for all 4 of these. So here is what we do. We have the RefMed database of names, which can handle all these 4 groups that I just mentioned. The top 2, groups 1 and 2, have defined structures which we can relate to a structure database, and there are other metabolite structures in there. Now these 2, groups 3 and 4, these are only partial structures in some composition. So there are no distinct metabolite structures associated with them, only the names. Another way of looking at this data is looking at RefMed. It's a database of names. It's about 170,000. About 25,000 of which are defined structures in group 1, group 2. The rest, a large number of these are lipids, cannot be defined as exact structures, but we do have harmonized names names for those. So it's important to see the distinction here. I mentioned this in our last slide, how we classify all of these metabolite names, whether they're lipids or non-lipids. And we have 3 levels of classification: superclass, main class, and subclass. There's a section on the workbench in the databases section that covers RefMet. You can search, you can download the entire set of names, 170,000 approximately. We have a publication on this. Here's an example of a detailed view of a RefMet metabolite. This is cholic acid. It gives various information, systematic name. One can choose an adduct, let's say M plus H, M plus NH4, and calculate the M over Z value from the neutral mass. You can look at other enantiomers of this metabolite. We have links to reactions in human pathways for this molecule, cholic acid, and so on and so forth. And up here, there's a link to the Metabolomics Workbench structure database, which gives more information. So here's an example of the different enantiomers and diastereomers for cholic acid. So these differ only by the stereochemistry for cholic acid. So we have 3 of these, or 4 of these for cholic acid in total. Very importantly, how can you convert your own list of metabolite names? Let's say you've done a metabolomics experiment and you want to convert your own list to the RefMet format. Then we have a convenient form where one can convert that list of names to RefMet. So the output will look something like this. This is the input name on the left. The standardized name, the RefMet name, is in yellow. There may be some that cannot be converted. So on average, about 90% of all metabolite names that we get in NDR can be converted to RefMet. The others have not been entered in our database yet. So that's how you actually convert your names. And
hi, Owen, let me interrupt you here. So someone has a question. Let me allow him to talk. I think it's David from Stanford. David, see if you are able to talk now.
Yeah, so can you hear me?
Yeah.
Hi, Owen. So I'm David from Motorpack Stanford. So maybe you explained to me this before, but why did you guys decide to create a new a standard of a name nomenclature for naming the metabolites instead of using, for example, KEG? What is the rationale behind that?
There's a very good reason, David. So let me go back to here. This is why. KEG or PubChem or any other database that you can name will not be able to handle all of these 4 different levels. It could handle level 1, exact structures, and possibly level 2, but it's useless, absolutely useless for level 3 and level 4. We get huge numbers of metabolite annotations at level 3 and level 4, where the exact structure is not known. So that's the main reason why these will not work. And that was the first thing we thought about way back when, but they will not work at all. So that's why.
All right. Thank you.
This is a very important slide. You really have to grasp this to grasp metabolomics. It's not about exact structures. It's about partial structures as well. Okay. Depending on whether it's a targeted or untargeted analysis, is really important to grasp this.
And there was one more question by another attendee. Looks like it's NG Enko Shawan. Their question, they tried to submit a study, LCMS message study, but they had some confusion about the study design. So if time permits, you can briefly go over the study submission page, maybe. And I suggested that if we do not have enough time today.
Yeah, we can do that after, yeah.
If needed, they can also contact us through the contact us page on the Metabolomics Workbench.
Yeah.
All right. So maybe we can give another minute for any additional questions before you proceed.
All right. Okay, I'll continue on. And like I was saying, this form allows one to ad hoc convert metabolite names to RefMet names. Now, in the study submission scenario, the NMDR does this for you. So we will automatically convert all your metabolite names to RefMet names. So if you look at any study of named metabolites, you'll see two columns, one with the submitters' names and the other with the RefMet names if they're able to be converted. Another feature you can use is to perform an IONATA calculation at the same time as converting the names to RefMet. So in this example, the submitter is selecting M plus H. So it standardizes the names and also converts all the neutral masses to M over Z for the M plus H adduct. So that's another thing you can do with this. Recently, we've also included mappings to HMDB, PubChem, KEGG, lipid maps ID, KEBI ID, and INCHI key if they are in the RefMet database. Now, you can't just put in any PubChem CID of the 100 million that are available. If it happens to match a RefMet name, then it'll be mapped. So down at the bottom here, you can see some of these have been mapped to RefMet. Okay, let me get rid of this thing. So like I mentioned the last time, we also have a RefMet classification where one can browse the RefMet classification by superclass, main class, subclass, look at the core structures. So if you want to know what acridone and anthranilic acid looks like, you can use this part of the website. And we have actually a database of core structures where you can search for all of these structures. Many of these classes are very similar in terms of name, especially in the flavonoids: flavanol, flavanone, flavanol, flavan. So it's good to have a reference, easy reference, to be able to look at these structures if you're interested in those. RefMet also has a number of OR packages where you can build in the OR mapping infrastructure into your own workflow. So there's an OR package on GitHub. There's other OR scripts and Shiny apps that you can use. There's even what we call a phone app that you can use to do that. This is not actually a phone app. It's a web page that's designed for using on mobile devices in terms of sizing. So it acts just like a phone app. So in summary, the RefMet maintains a very central role in terms of standardizing names, very important, obviously, in comparing and contrasting studies. Unless you have a similar name to map with, then you can't compare and contrast data because of the heterogeneity in terms of synonyms. It's connected to a structure database for those RefMet names that are exact structures. Classification is very, very important in terms of doing, say, graphical analysis, statistic analysis on subclasses, main classes, reporting, and so on, and connecting with biochemical pathways. So what are the positive outcomes? Unifying nomenclature. We absolutely need that in metabolomics. We absolutely need that. This is one of the most important things that we need to have to compare and contrast data to report molecules that are easily interpretable. We have connections to exact structures to databases in the cases where they are exact structures. We have a classification system that has numerous advantages in analysis. And very importantly, integration with biochemical pathway tools. You'll hear about this more from the talks in the next couple of days. We also have a REST service where one can look at the data on the workbench in a number of different contexts. If you're interested in metabolite structures themselves, there's a number of features here to do that. If you're looking at studies in an MDR, there are a number of REST services to do that. MetStat refers to a summary method that I'll talk about more in detail. You can do a RefMet search, download through REST. We have a gene protein database, and also you can do mass spec searches through the REST service, which has its own API as well on its own page. The metabolomic structure database, now I talked about that in a few slides back. So this is a database of exact structures, some of which are in RefMet, about 25,000, and then the rest then are from taken from other major databases like PubChem, KEGG, KEBI, HMDB, and so on. So one can go into search this by text, by substructure with the substructure drawing GUI, and so on and so forth. You have a detailed page. Importantly, for every metabolite structure, it gives links to the studies where that structure is found in using the RefMet mapping. So, for example, 10-hydroxycapric acid has 34 different studies where that molecule is actually found in. This is the detailed view for one molecule, cholic acid. It gives all the database links that we have for this particular molecule, human pathway links as well. We have omics integration, in this case, mapping to human metabolomics pathways, either using HMDB and KEGG pathways or reactome pathways. So again, one can jump from metabolite to one of these constituent pathways that molecule is found in. We also have an in-house gene protein database. Now, this is something entirely different. This is we're talking about genes and proteins here. So this database is comprised of genes and proteins that are known to be related to human metabolism, going through various entree categories and so on and so forth. So this has about 7,300 genes and over 15,000 proteins. You can search by entree gene ID, the gene symbol, if you know that, or by pathway. And so here's an example of the MGB database. It's also linked to the metabolites in that pathway. So that's a way of getting from the gene pathway to the metabolites themselves. And also the panel here on the bottom right shows one particular HMDB pathway that all of these molecules are matched to. So this is the detailed view for a gene protein record. In this case is mitochondrial aconitase 2, gives a gene symbol, the entree gene ID. And here's the detailed view of the protein. So this one has precursor protein. It's got a transit peptide. So the mature peptide is shorter by this leader sequence, which is clipped off going into the mitochondria, and links to all the other gene IDs and mRNA IDs as well. So that concludes this talk. I'm going to talk about analysis tools after we have a little break here. So this is a good opportunity to ask some more questions if you have any questions.
So there are two questions. So maybe you can go to the question and answer panel as well if you like, or I can just read the question.
Okay.
So one of the questions is from Sega. Could you explain the differences between the main class and superclass of RefMet? Sometimes the main class and the superclass are the same.
Oh yeah, so the superclass is the highest level one. So for lipids, it's fatty acyls, glycerophospholipids, glycerolipids, nucleic acids, carbohydrates, benzenoids, and so on and so forth. So occasionally, the subclass and the main class may be the same, or maybe even the superclass and main class are the same. These ones are derived from the classifier classification. This is a method developed a few years ago at the University of Alberta by David Wishart. And it's not perfect. It takes on a very, very tough task in trying to classify all known metabolites, and there are literally millions of them at this point. So it does that by structural logic, and in some cases it's not very discrete in terms of having enough granularity. So in cases where you're looking at one particular superclass that doesn't have very much granularity, then maybe the main class and subclass are the same. But for all the more heavily studied metabolites, this hierarchy is very, very good, I think. It's just more the outliers, more the esoteric ones that don't have that level of granularity. And again, it's a matter of how much work you can put into this. If you have tons of curators, then you can have a more detailed hierarchical scheme. But we try and handle most of the commonly encountered metabolites and do a really good job on those.
So the second question, actually, I think it sounds more like general information. Dr. Ashok Gehlot says, MW has reference data on the metabolomics of plants and the biochemical pathways involved in the production of disulfides. So I think he's referring to his previous question related to sulfur metabolism.
So in terms of pathways, we've really just focused on human pathways. It's just a matter of resources, really. There are other organizations and efforts that out there that put a lot more focus into plant pathways. We're just using KEGG, HMDB, and Reactome to link to their human pathways. It's really a matter of resources. I mean, don't get me wrong, plant pathways are very, very important. It's just a matter of the amount of people we have at UC San Diego to work on this. It's a numbers game.
There is one more question in the chat by Sunita, and the question is, if we have screened a set of metabolites and I wanted to check if that could be a biomarker for a disease condition, is there any option in a Metabolomics Workbench to identify that?
So what you could do if you're interested in one particular disease, or for any disease, it depends on the approach. If you're interested in one particular disease, you can very quickly ascertain which studies have submitted work on that particular disease, and look at those studies. Another way to do it, probably a better way, would be to search for that metabolite, especially the RefMet name, in the workbench. Just do the quick search and get that list of studies. Let's say it's 45 different studies. So those will be listed by the study title. And if any of those refer to the particular disease state that person's interested in, that would be a good way to do it. So that you would narrow it down to that. But then you'd have to look at the details, exactly, has this metabolite been up or down regulated in what types of samples? So it's kind of a big question, but that's the way I would tackle it.
Yeah, I think once you identify a couple of studies related to the disease of interest, going into the details of the analysis tools such as the statistical analysis, look at the t-test or p-values and the ones which are showing a strong p-value, go further after those in terms of identifying what pathway they belong to and what are the literature they have been cited to. So I think that could be a good starting point.
Like I say, in summary, there's two ways you can do it. You can go from you can start from all the studies for that particular disease associated with that disease, and then look at the metabolites, or start with a metabolite and find all the studies that contain that, measure that metabolite, and then break them out into different diseases based on the information in the study title.
I think we can wait another 2 minutes for any additional questions before we take the break.
There's an additional question, Mano, in the chat.
Okay, so this question is from Jane. Is there a way to Venn diagram the potential source of secondary metabolites in, for example, blood, such as bacterial toxins, plant metabolites?
Venn diagram, let's see. I mean,
okay.
So if you have a blood sample, what you're measuring is all the metabolites in blood using whatever detection method you're using. Let's say it's LCMS. You know, you might do it by reverse phase LCMS, Hillick MS. You're not going to know a priori where these are coming from, but it all depends on how the study design is set up. If the study design is set up where the investigator is using has identified various bacteria in those samples, and so maybe you have positive, negative controls, then you would be able to differentiate between the samples, whether certain metabolites are coming from the bacterial source or from the the human blood source. It all depends on how the study design is set up. So obviously that's crucial. You have to look at that.
Well, I guess where I'm getting at is maybe a couple different concepts, and that's that if there's a fingerprint for certain bacterial metabolites already, then one could do some
okay, I see. Yeah.
Right. So
Gotcha, yeah.
for example, like I don't know how many samples you get from microbiome studies, for example.
Yeah, we're starting to get a lot more. Yeah, from gut samples. Yeah, this is a pretty hot area now. Yeah, very hot area. Yeah, we do quite a few.
So how are you going to be able to go over that at one point in this, to see how to search for that kind of stuff?
You can search for individual metabolites. Now, are you talking about patterns?
Sure. Correct.
Yeah. We don't go into that level of detail because that's it's a very, very specialized case. But I understand what your question. That's a very interesting question to be able to profile these for the presence of a particular microbial metabolite.
And I guess the other question would be if you can link, sorry for interrupting, you're going to say, but to link them to disease states as well.
So you're saying if it contains certain metabolites, then it links to a certain disease state?
Oh, no, I'm just asking. I think you're going to say it depends on the study design.
Yeah.
Like I could look it up based on a study, but it wouldn't be an unbiased search saying, hello, I want to look at blood and then I want to further capture here's blood from a disease state versus a normal state, and then let's see, kind of correlate those metabolites and say, hey, here's these secondary metabolites, kind of a Venn diagram saying these are potential, they've already been characterized in bacteria as commonly secreted by bacteria, pathogenic ones and normal ones and things. And you can kind of, that's more
No, it's a
Where-
very relevant topic because we have a collaboration with the research group at UC San Diego, Peter Dorstein, who's doing exactly this. They're going back and looking at Crohn's disease and various gut diseases by synthesizing all of these bacterial bile acid conjugates
Yes,
and
yes.
reanalyzing data that previously showed up nothing, and being able to discriminate between controls, Crohn's disease and IBD using bacterial profiles from these molecules. So yeah, people are doing that, but of course they have the expertise in terms of the biology behind that too.
Sure.
It's a very interesting area, and it's really taken off. Yeah.
But as far as the database, as far as unbiased searching right now, I'm just wondering kind of where it's at or what the plans might be for future.
So what you can do right now, if you have a study that is looking at bacterial metabolites, you would use the study design. Let's say you find a study of interest, then you go to that analysis portal. I'll talk about that more in the next talk, and compare and contrast controls versus Crohn's disease samples or whatever. And we have tools to do that on the workbench, where anybody can use that. Of course, if you have expert knowledge in that area, you can go much farther probably by just downloading all the data and doing your own analysis. But yeah, there are tools to do that, to compare and contrast and see which metabolites are up and down regulated across different samples.
But then it's hard to know where some of the sources are because there's I can't do I couldn't say, for example, do a bioinformatics between, okay, now here's because it's hard to correlate unless you know the microbiome and you know what species of bacteria are there to begin with. It's such a complicated open source to even know.
Yeah, I think you need expert knowledge to determine exactly which bacterial species you're dealing with.
Right.
I think that would be beyond the scope of Metabolomics Workbench, where we're basically our job is to be a repository and to develop generic tools. But at that level of specificity, then it's better to download the data yourself and do your own analysis, I think.
Right.
Yeah.
I just wondered about how much data was stored in, for example, the bacterial or plant, and how I could go there and what I would see.
So if you have the expertise, you could download the raw data, the raw mass spec data, and do your own analysis. So that will be probably the best way for people who have the expertise in mass spectrometry and downstream processing. Yeah.
Okay. Thank you.
Owen and Jane, if I may interrupt, I think it might be worth visiting this question after Owen, you have presented about Metastat, because I think that provides filters for disease, species, class, and so on.
Yeah, exactly. Yeah, we'll go through that. So what I want to discuss now about how to look at summary statistics from NMDR. So as I mentioned, we have over 3,200 studies available. So what we've done is we've coalesced all of these into various types of summary reports where you can ask questions. For example, if you're interested in a particular analysis type, you're involved in LCMS and positive ion mode, you want to see what metabolites have been discovered using that analysis techniques. If you're interested in disease association studies, obviously, if you're interested in species or sample source, for example, you want to look at human liver. If you want to look at one particular structure class that you're interested in, if you want to ask questions about what metabolites are detected with selected criteria, which metabolites show significant changes across experimental conditions, which biochemical pathways are these metabolites involved in. If you want to look at variance across sample replicates for a metabolite, or look at ANOVA p-values. We have a system called MedStat, which basically summarizes all of the data for named metabolites on the workbench using the RefMet nomenclature system. So we'll look into that in detail. So there are 2 links to MedStat. This is from the Browse and Search Studies portal. You can, the top one is the most simple one. You view the most frequently encountered metabolites in NMDR across all methods. And then the bottom one then, where you can filter the data and look at much more detail. So the first one shows all frequently reported metabolites across NMDR studies. So this changes by the day as new studies get added. So it's essentially a big long list of RefMet names, with the topmost one being the metabolite that has been found in the most studies. So for example, no surprise, most of the top hits here are amino acids. So glutamic acid has been found in over 1,100 studies, and so on down the list. The colors indicate the the superclass. So pink is organic acids, amino acids, and stuff. Green is nucleic acids, blue is carbohydrates, and so on and so forth. You can click on those links to get the list of the studies in the second column. Or if it's an actual metabolite, you can click on the metabolite link to go to the structure database. So you can search, this is by default 200 records. You can search all of them, which is a very, very long list of thousands of different metabolites. Notice that the names are all pretty simple. This is the premise of RefMet, to use the simplest name possible so that people can fairly easily identify what it is. Now, the more detailed approach is you use a query builder. So if you were interested in a particular analysis type, chromatography type, and so on and so forth, if you want to look for all metabolites found in human blood, you can do so. So this GUI is set up to look for all metabolites in human blood. So you click on that link and you get the list of all metabolites found in human blood. So again, it's a very, very long list. You can link to structures, pathways, lists of studies. This value here, the RSD, is the relative standard deviation. It gives an indication of the scatter. Now, the RSD is calculated separately for each experimental condition within each study. So if one study has like 10 different experimental conditions, groups, i.e. groups of samples, it'll be calculated for each and every one of those and then averaged. So you can do various things with the query builder. If you're interested in looking at metabolites detected by HILIC, LCMS in negative mode in blood, you're going to get a very different profile than you would if you did the same thing for example, LCMS in reverse phase. So you can get, so the advantage of this is that if a researcher is going to look at some particular tissue and some particular organism with a particular analysis method, let's say LCMS using HILIC or reverse phase, they can look at all of the studies that have used that exact set of criteria and see what's being detected so far. So that gives them a very good overview as to what to expect and what can be detected using those criteria. Here's a different one where the key identifier here is the human pathways, the citric acid cycle. You're just looking at citric acid cycle metabolites detected in humans and by LCMS in negative ion mode. So again, you get a very, very different profile, and it gives the numbers of studies here that have been detected with that method. And here's another example. If you're looking at one particular metabolite superclass, you just want to look at phospholipids and nothing else, detected in negative ion mode by LCMS in humans. Again, you get a very, very different profile. So many of these now, almost all of these are not exact structures. They're molecular species, where, for example, only the total number of carbons and double bonds is known, and the head group is known. So MetStat also links to the data itself. So, for example, in this case, a valine, it gives you a histogram of the RSD values, and it gives you the median values of the RSD. So the closer this approaches a bell curve, it gives more of an indication of, first of all, the numbers of studies, and also the reliability of the RSD value. It also gives more information down below on the ANOVA p-value for that metabolite in a particular study, depending on the conditions. So these large p-values indicate that this metabolite varied hugely across these different criteria. Of course, you'd have to look at the actual study to get more detail about that, but it gives you an indication of what metabolites are changing the most across different conditions in a particular study. Also, you can get information on the quantitative data. So, for example, for the amino acid proline, it lists the table of results, all the studies. On the right-hand side here are units. So it's very important to choose quantitative unit like micromolar, not counts, because these are the ones that would be targeted data. So, for example, if you click on for micromolar here for proline, it gives you a box and whisker plot of proline across all studies, across one particular study in orange. And then down below, you get a comparison across all studies for proline. Now you notice that the outliers are very large. So probably one particular study had some extreme outlier, maybe due to the conditions or maybe due to the way that proline metabolite was measured. And here is the corresponding box and whisker plot for one study. As you notice, the medians are pretty close together. So this gives you an example of the typically measured concentration of proline in human blood across all our samples in terms of micromolar. So that's a way to get information on quantitative information. Switching gears now, another tool we have in the tools menu is to do MS precursor ion searches. So one can copy and paste a list of precursor ions, MS1 data in here, or upload a peak list file. Then depending on whether it's positive or negative ion mode, choose various adducts to search for. You also have to decide which database you're going to search against. Are you going to search against RefMet, 170,000 metabolite species, virtual database of lipid classes, lipids only, generated computationally, or the MW metabolite database, which includes the discrete reference structures and then a lot of other structures like drugs and other molecules. So you run your search, get your results. If they're linked to exact structures, in the case of RefMet, you can click on the structure link there. So you can also search untargeted MS data by M/Z or retention time. So as I mentioned, we get a lot of untargeted MS data saved as M/Z retention time pairs in sets of files, which we can also upload to the database and use that as a search endpoint. So for example, you can search, I think it's about 5 million M/Z retention time features across untargeted studies on the Workbench for a particular M/Z value, set the M/Z tolerance. If you have retention time information, you can also limit that to the retention time and tolerance too, and further optionally specify instrumentation. So for example, for this simple search on 657, it gives the following results. And as you notice here, so these are all the matches to that M/Z value within that tolerance. As you notice, there are a number of other annotations here. These are named metabolites for these diacylglycerol species in this case, where the M/Z value was also supplied. So these are indications as to what these M/Z values might be, but you also have to take into account the retention, the chromatography information as well. This is all in positive ion mode. And you can get more details on the actual analysis conditions by clicking on the analysis link here. Another set of tools we have are structure drawing tools. So these cover various types of classes, acylcarnitines, bile acids, glycerolipids, glycerophospholipids, ceramides, sphingomyelids, and so on. And the further resources, an excellent resource here is the Lipid Maps website, which has its own set of comprehensive structure drawing tools for lipids. So, for example, one can draw sterile structures. You can select between 45 different sterile cores up here. Basically, add your functional groups, specify stereochemistry, double bond position, draw the structure, and it will also generate a systematic name. You can calculate the M/Z value by selecting this pull-down menu and so on, and download it as a MOL file, where you can enter it into other software or download it as an image file. For other lipids and molecules, you can select between 65 different head groups here, and there's a big long list of these are lipids, and then you can specify what the SN1 position, SN2 position, SN3 position is for phospholipids, or for ceramides, what the sphingosine base is and what the N-acyl chain is, and draw the structure that way. Again, download them and save them as PNG files. So it's just a useful tool to draw structures in a controlled fashion. Another aspect that's quite important is the toolbox for individual studies. So if you go to any study of interest, let's say study 1140, go to the main page. There's a link called Perform Statistical Analysis. You click on that and you get the study statistics toolbox for that one particular study. So it looks like this on the right-hand side. So it covers various categories of analysis, metabolite classification by RefMet in terms of pie charts, normalization and averaging. So sometimes our data is already normalized and averaged, but one can do that using tools on the workbench too. You can normalize by sample normalization or do analyze scaling. These are very common techniques for post-processing data. You can do various univariate analysis, especially volcano plot is very useful and NOVA is useful. Meta-EMP, we'll talk a bit more about that tomorrow and the day after. You can do clustering and correlation, multivariate analysis like PCA and LCA and LDA, and then do other classification analysis like random forest, and also map to human biochemical pathways. So all of these links, like I say, are available for that particular study. So for example, and one other feature is you can do molecular structure similarity analysis. So what this does is it takes all of the named metabolites for a particular study, or you can do this ad hoc as well, and convert them first to RefMet names, and then look at the actual structures, if the actual structures are known, and do a correlation, structure similarity analysis. This is a fingerprint similarity analysis using Python software, where you're looking in this case at just the structures. How similar are the structures? you can map those onto the subclass. So each of these colors here represents a RefMet subclass. Now, as you can see, certain subclasses are closely correlated by structure. So, for example, on the bottom left panel, for example, the ether LPCs are very closely related to the regular LPCs because there's only a fairly small structural difference, and so on and so forth. So it gives a good overview of the structural similarity for a particular study set. So this is a study analysis toolbox. There are many different options here. One option is to look at pie charts of metabolite classes. So you can look at the superclass level. So you have organic acids, fatty acids, the major lipids, carbohydrates, nucleic acids. You can drill a little deeper and go into the subclasses. There's also a main class where you're looking at much more detail in terms of the structures. Phosphatidylcholines, triacylglycerols, hexosylceramides, and so on and so forth. Or you can limit that to one particular superclass, let's say glycerophospholipids. So now you're looking at all the different glycerophospholipids subclasses only for this particular study. So it's a good, fast way of profiling entire study. Normalization and analysis, you can perform that. Obviously, samples should be normalized. Basically, what you want to try to do is get the same total intensity for every single sample, because to protect against different injection variability and stuff like that. You can do that. You can also do analyte scaling. So some metabolites like cholesterol are detected in huge amounts, and maybe other metabolites like desmosterol or other sterols are much, much lower. So you can scale on the analyte dimension to reduce the huge variability there by using log methods and so on. Another thing you can do is cluster the data. So you can, in this case, you're clustering in 2 dimensions. On the top here, you have the different conditions for that particular study. And in this case, I think the samples were treated with 2 different drugs for 2 different time points. So you have 4 different conditions. So you're clustering in two dimensions, both in the measurements in the metabolite dimension on the y-axis, and also across the samples on the x-axis. So you can do a heat map cluster analysis like that for that study. In terms of univariate analysis, we have a NOVA tool where you're looking at the analysis of variance across the different factors, a different set of samples. So, for example, you have a sampling time point, or in this case, you have a drug treatment, and you can run ANOVA on those samples. And these are the ANOVA results. They're come in 2 tables. Since we can group all the names into RefMet subclasses, you can look at it from a subclass standpoint, which subclasses are most highly differ between experimental conditions, or more commonly, you just look at individual metabolites, which p-values are highest for which metabolites. On the right-hand side, you just have the same data in a graphical format. It's basically the key metric here is the -10 log p-value on the y-axis, and the classification index is just spreading out the different subclasses, so you can see them more easily. Another, probably the most commonly used analysis tool is a volcano plot analysis. So now we're comparing 2 different conditions, 2 groups. So in this case, it's a time-dependent duration. These samples are treated with a drug, glucocorticoid, at 2 different time points. So you're looking at the 2 different time points. You run the volcano plot. You can run it on all analysis for this study. This study had 4 different analyses. So you can group them all together or look at them one at a time. You set the p-value, fold change, cutoff. These are standard volcano plot parameters and so on. And you run your volcano plot analysis. So again, you get information on the individual metabolites. You got the volcano plot, which is probably the easiest graphical method to look at it. You can look at the data also by enrichment analysis. So looking at it in more detail, again, you can look at metabolite subclasses, which subclasses show the greatest fold change and greatest p-value, or look at it by individual metabolites, which show the greatest p-value and fold change. So the standard way to look at this is a volcano plot where you have a fold change on the x-axis, either in the positive or negative direction, and t-test p-value on the y-axis. So the ones up near the top corners here and here tend to be the most statistically significant. And this gives you the metric of the fold change. And there are a number of ways to look at this data: by bar plot, by class enrichment plot, if you're looking at entire subclasses. We can do network analysis tools using various correlation algorithms. One is the Pearson correlation, very well known. There's another one called DSPC, developed by the University of Michigan, one of our collaborators, which is more powerful for sparse data. So basically, you're looking at how metabolites are correlated. And the output is this graph here, where each of these colors represents a subclass, and the connections are, the dots are the nodes, the metabolites, and the edges then are the correlation values. So you hover over one of these lines, these edges, to give the display the correlation coefficient. You can filter by subclass. So for example, if you just want to look at cholesterol esters, you just click on this link here and it only shows the cholesterol esters. You can also map you do the same type of analysis and map it to fold change as opposed to classification. So now you're looking at the up and down regulated metabolites, significantly up and down regulated metabolites, green and red. Up is red, down is greens, to give you an indication of which metabolites are moving in the same direction. And going to the multivariate analysis tools, most of you, I'm sure, have heard of PCA and LDA. You can run LDA, for example, on that study. We're in the study toolbox. You select your number of conditions to analyze. In this case, all 4 different experimental conditions were selected, and 4 different zones are found in the scores plot corresponding to the 4 sets of samples. You see the loadings plot here, which indicates which are the main drivers of the LDA, as well as a graphical output of the significant loadings in the 2 principal component dimensions. Also, you can map metabolites to human biochemical pathways. Obviously, this is more relevant if your samples are human samples. So again, you can map your data, and the output is down on the left-hand side, where it shows which metabolites are connected with which biosynthetic pathway and which are have the highest t-test value and so on. These are linked to SMPDB pathways. This is the HMDB pathway system. And you can also get a pathway enrichment score, which shows which pathways overall are most affected. We have also, I want to mention meta-analysis tools. Now in this case, we're not looking at one study. We're looking at 2 or more studies. So for example, you can look at metabolite ratios across, say, 3 different studies, where you have a GUI which allows you to select 2 different metabolites that are present in all of these 3 studies and look at the ratios and graph that. Another important tool is to be able to compare metabolites in 2 selected studies. So any 2 studies that you wish to, that you're interested in, you select those using this GUI, study 1, study 2. It generates a Venn diagram which shows the overlap, if any, and it shows all metabolites in study 1 only, study 2 only, and metabolites that have been found in both studies. So again, that depends on the RefMet nomenclature to be able to link across different studies. One other thing you can do is analyze your own data set. Let's say you don't want to submit a study. You have a data set in a table format that you want to run some of these statistics tools on. You can do that. So you go to this GUI where you upload your own data. You can also view the example file. I would do that first just to see what the data looks like. So the data has to be in a certain format. So in column 1 are the sample names, and then column 2 is a group identifier. So that could be control, disease, time points, whatever it is that's different about the samples. And then all the remaining columns then have headers for the variables, typically metabolite names. So this is an example of a test data set. So in the first column is the samples, the second column is the group, affected male, affected female, control male, control female, and then all the different columns for the different metabolites. And then you basically map that to RefMet. You don't need RefMet, but it allows you to access some other tools, and then you run your samples. So here's an example of the tools that are available to you once you've loaded your own your file of data: normalization, scaling, bar plots, univariate analysis, basically all the tools that I've just described to you a few slides back. But the difference here is that it's just it's the user uploaded data. It's not a study. And again, you can have all of these are just examples of all the things you can do with loading your own data set. So it's just a quick way of being able to access the analysis tools on your own in an ad hoc version. So I want to close by discussing how NMDR on the Workbench maintains fair principles. Obviously, data needs to be findable. So when a submitter submits a study to the Workbench, it's to NMDR, and it's being finalized and curated. The submitters are assigned a DOI. So this is an identifier that can then be used in publications. So it's something that's not going to change over time. It's a link to that project. So if you have multiple studies in one project, it'll link to all of those studies in that project. It's shared with various other global repositories like EBI, Omics Discovery Index, and Metabolomics Exchange. These are global repositories that save data from metabolomic studies around the world. So not just NMDR, but also metabolites in Europe and other sources. So one can go to those resources and find metabolomic studies across all of the repositories. Accessibility, we've talked about that. Multiple browsing download options through the Workbench, and the REST API is very powerful for accessing data in computational mode. Interoperability is very important. We have the MW tab format, that text format to describe the data, which can be used and shared so people can develop their own computational infrastructure that will work on MW tab if they so desire, which contains all of the key metadata and named data components. And obviously multiple positive outcomes via RefMet, the standard nomenclature. Let's go back one. Reusability, this is huge. It's so important to have raw data available because instrumentation methods and lists of standards are improving all the time. So one can go back, in future years and reanalyze existing data and find many new metabolites because more standards are available or more in-depth processing workflows are available. So this is very important to have this raw data available, and this is why we push it so hard. So I'd like to thank you for your attention. I want to point out that this is a NIH Common Fund project. It's been ongoing since 2012. I'd like to in particular thank our curators. Neil and Shashi have done tremendous work curating all of these studies over the years, and also Shankar Subramaniam, our PI on the project at UC San Diego. So with that, I'll conclude and take any other questions or anything that people want to talk about.
So, and there is one question, and it is related to the factors. So the question is it okay to group factors into a single factor column in study design, or is it preferable to search each factor into its own column?
It's much more preferable to do it into its own column, because then you have further granularity on how you can analyze the data. So let's say you have one factor for disease type, control, test, another factor for treatment, another one for, say, time, analysis time or whatever, or treatment time. If you separate those out into different factors, then one can look at each of those experimental conditions individually. So that it's much more powerful to do it that way.
I think the remaining time till 11 a.m. PST. If attendees want to explore, go to the website, Metabolomics Workbench, and explore different sections. And in the process, if they have any questions, they can ask. And I recall there was one question related to clarifying some things on the study design as part of the metadata submission process. So, website.
Yeah, we can go over a few things here. I want to just bring your attention to some key areas that are very, very useful. Obviously, the quick search up here on top, it's found on the top most pages. So you can search for, a particular metabolite, for example, citrate. Okay, oops. Okay. So I typed in citrate there. Okay, so the proper RefMet name is citric acid. Okay. So let's say if you want to, the first match here is partial name search citrate in RefMet. So there's a metabolite called monoglyceride citrate in RefMet. The more relevant one is citric acid, which is probably what you're looking for. So this is the RefMet page for citric acid. So for example, you can look at different adducts for that citric acid. So this would be relevant for what you're likely to detect in a mass spec experiment if you run it in negative ion mode, okay? So we have various information on citric acid there. Now, since this is an exact structure, you can link to the structure database. Now you're in the metabolomic structure database for citric acid. Gives the systematic name, classification, various other mass spectra. This is from MassBank. Various other database links, PubChem, CID. So this is a very, very well-known molecule, obviously. So it's available in a ton of different metabolite databases. So let's back up here a bit. So citrate, the name citrate is found in many other metabolite names. in our main structure database. And so these are all the ones you see here, and you can click on the different structures if you want, get information on those. And these are the studies that have contained the word citrate in them for a metabolite. So this monoglyceride citrate was detected in those 3 studies. Now, if you wanted to, studies with citric acid, it's probably better to type in citric acid. Now, 890 NMDR studies have reported citric acid, not surprisingly, since it's a very common TCA metabolite. So I just wanted to go over that. I use this all the time to find a particular study of interest. It's case insensitive. So this is probably the fastest way to find a study of interest. So this is study 1140. So this is what the main page of a study looks like. You can look at the samples. So this is a very good example of the study. They've broken out the experimental conditions into treatment group. So they have 2 different drugs. This is a study on dogs. Treatment duration, 0, 4 days, and to the other one, 0 weeks, 25 weeks. Also sampling type before and after, well, that's the same. So, and they have other metadata too for the samples. So these are 3 key study design components where you can discriminate if the data is samples are set up like this, then you can compare 2 different drugs, for example, 2 different time points within a drug, or you can compare and contrast any of these 3 different features. So if you want to look at named metabolites, in this case, I think there are 4 different analyses performed. So these are metabolite names given by the submitter. These are the RefMed names, standardized names, and big long lists. This is the second analysis. They did a separate analysis for sphingolipids. Again, the metabolite names and the RefMed names. And they had 2 other further classes, sphingosine 1-phosphates, and also triacylglycerols. So you can look at one particular metabolite of interest and get all the measurements. These are in micromolar. So this is a targeted analysis showing the different factors. These are the different samples and the different measurements. So you can do bar graph of all the samples. You can do a box plot of the samples. So this is the average across all of them with the median. You can do it by different factors. So this is probably the most interesting. So you have 4 different groups here. You have 2 different drugs, 2 different time points, and these give the box and whisker plots with the median values. So that's probably the most useful part. You can download metabolite data in a table. So if you want to do your own analysis, you just click on these links here and you get a table of data. You can download the MW-tab file. So like I mentioned, there are 4 different analyses. So let's look at what an MW-tab file looks like. It looks like a big text file. So divided into different sections: project section, study section, the subject section. So this is a beagle dog sample with the common name and the Latin name. There's a subject sample. There's a study design section, very, very important in certain format. Then you have collection, treatment, sample prep, chromatography, what instrument name, column name, et cetera, et cetera. Now, I should mention all of this data is also absorbed into a relational database. So mostly when you're looking at on the website, you're looking at the relational database. but I'm saying you can download this MW tab file and do your own analysis. The MS, there's an MS block on how the study was acquired, and this is the data. Then this is the actual data table, samples, factors, and measurements. So you can do that. You can also download it in JSON format if you want to access that format. And then very importantly, you can download the data files. You can view the archive contents. So these are all the actual files, all the raw files. So these are .d files in this case, adjuvant files. Ideally, we like them in open source format like MZML, so on and so forth. So another important link is to perform statistical analysis. So this is what I was talking about, for the previous 45 minutes. All of these classes of analysis that you can do. So for this particular data set, you have a choice of 4 different data sets. So if you want to look at the pie chart of superclasses, there you go, at the highest level. Main classes, you're drilling down to more detail. And finally, the subclasses where you have the most amount of detail. So, so that's how you do that. Let's look at volcano plot analysis. So you have a GUI here where you set up what you want to compare and contrast. So let's say we want to look at one drug treatment before and after. Okay. So group 1 will be before, group 2 will be after. Okay, p-value cutoff. So, well, we can leave it as a default. We can do sample normalization or not. You can select classification by subclass or main class. You have a choice here of using the submitted names or refmet. And let's just use submitted names. And importantly, we can combine all the data for all the analysis. Let's do that instead of looking at each one individually. Okay, so now we're going to compare prednisolone treatment before and after, all right. So you run the volcano plot. Okay, so it just finished now. And here's the tabulated data results set. We have one set for, let's see, so we have one large table which shows the t-test p-value, the log 2-fold change. You can sort by t-test. So these are the most significant t-test values by metabolite class. And down here, then you have the t-test p-value for all the individual metabolites. So lots of glycosphingolipids, hexosylceramides, phosphatidylinoxidols, and so forth. So the standard way to look at a volcano plot is like so. So you have these phospho PIs up here in the top left corner. That means that they have a very significant t-test p-value, and also the fold change is quite large. And then on the other side here, these are the upregulated ones. You have a lot of glucosylceramides and cholesterol esters, again, very significant t-test p-values. So it's just various ways of looking at the data. This is class enrichment. So which classes, in this case, subclasses, are most highly changed between these 2 time points. In this case, hexosylceramides, PIs, and PCs is telling you the same information that the volcano plot is telling you, but just in a different format. So let me go back a bit and go home. I want to point out a few things here. So we're in data repository, browse and search studies. Like bubble plots are a very easy way to look at data. So if you're looking at disease, species, human, mouse, obviously are the dominant one. Sample source, we'll look at sample source. So if you want to look at, say, urine, these are all the studies that contain urine as a sample source. Now they may have contained other ones too, and you see there are different organisms here. So it's a fast way of getting to the data for particular item of interest. The other thing I want to point out is MedStat. This is the summary view. So this is a study of all metabolites found in MDR, sorted by the metabolite that's found in the most studies. So here you have glutamic acid. You click on glutamic acid, then you get to the actual structural information, number of studies. You can change this to a large number. Let's say you want to see the top 1,000. You get a very long list. Goes on and on and on, colored by classification. So all of these are RefMet names. So that's one way to do it. The much more powerful way, though, is to do the detailed meta-analysis. So now we're going to do the same thing, but we're going to be much more specific. Let's say we're interested in blood. From, let's say, human. So these numbers here refer to the number of species that have data on blood. So these would be mammals. And let's say we want to narrow it down to LCMS, positive ion mode, using reverse phase chromatography. So now this is a fairly specific type of a query now. So now we're going to ask the question, show us all metabolites detected by positive mode LCMS in human blood. And click generate. So now we have smaller numbers of studies as compared to the global one. But again, you know, amino acids dominate. And it's a big long list of metabolites. So it's a very convenient way for somebody who wants to look at the field in total, if they have a particular sample and species, a particular analytic method of interest, to be able to see what's been found. So for example, proline, we get some data on that. This is the RSD profile for proline, and this gives an indication of studies where proline had a huge ANOVA value for particular experimental conditions. So here you're comparing blank case and control, so that's a very broad swath. Here you're comparing different, it looks like different extraction methods, methanol, chloroform, methanol only. So you'd have to go into the detail. Here's one for gender. So there's this much difference in this particular study between gender, male and female, and so on and so forth. And you can look at a totally different chromatography method then and get totally different results. So it's helic and negative ion mode in human blood. So now you have a much more, a lot more, several very hydrophilic metabolites as compared to reverse phase. So these are more water soluble. So I'll open it up at this point and ask if anybody wants to look at any particular other aspect on the website rather than me just picking and choosing. Does anybody have any questions?
If anybody would like to speak, please raise your hand, then we will unmute you so that you can speak. Someone asked question, are we recording the presentation today? Yes, so it is being recorded, and we will review it after the workshop is over in the next 3 days, and then we will post it after any editing.
Yeah, here's an example of a REST service. So this is available in the tools section. So widen this a bit. So like I was saying, there's different contexts here. There's a compound context. So if you want to select compound smiles from a PubChem compound ID, for example, you're inputting the compound ID, and it's reporting smiles. You can download, for example, if you want to study context, if you want to select summary information for a particular study. So up here is the study. This is just study 1. So it gives all the study summary. So you can do many different things. You can show metabolite name for an NMDO or metabolite ID. You can, let's see what else is. You can standardize a metabolite name to RefMet. So this one is standardizing citrate to RefMet. So you could embed this REST query in your own computer program and call it, and it'll do the mapping for you. So it's very powerful in that respect. You can fetch all studies containing a particular KEG ID. So this is in JSON format. You can also output in regular text format too. And you can look at particular gene symbol, for example. This is the gene symbol ACA. So if it's in the metabolomics gene protein database, it'll output data. You can search for a particular mass. This case, the query is what's the M plus H M over Z value for PC34:1? These are lipids. So it's multi-featured. It's the fast way of computationally extracting data from the workbench, and it has its own API as well. Sorry, I'm going a bit hoarse after talking for 3 hours. Anything else?
And one question was, if we have a pathway, can I find all metabolites in the pathway by this filter option? So I think this is from Silsa. I think the reference is to Metastat. So I believe it lets you select the human by the species and then the pathway name. So at least in principle, I think it should allow you to do that.
Yeah, there's various ways to do it. And there's also the human metabolome gene protein database, where you can look at different pathways there. And that's linked actually to metabolites too. So you can click on a particular metabolite that's present in that pathway and get information that way. And also there are tools in the analysis section where we can map data to pathways. Let's see. Go back to the one we were in. I'm going back to the toolbox. So you can map study metabolites to HMDB and KEG pathways. Okay, I got to be a bit careful about this. This is actually a dog study, so you're mapping dog metabolites to human pathways. I mean, it's pretty much similar. I'm sure the pathways are, but just be aware of the differences. So in this case, there are like 6 different pathways shown, and the ones in green are the ones that were detected in this particular study 1140. So only one of those were selected, and you can go to the actual pathway itself. This takes a minute to load. We're loading the SMDB pathway right now. This is on a different website. You can actually zoom in on it, and the metabolite of interest should be highlighted in red here. So it's a CoA. All right. Anything else? And Mano, you're going to talk in much more detail about pathway integration tomorrow with the CFD talk.
Yeah, I will cover some of that as part of my TNP.
Yeah.
I think it does not go into too much detail, but I think it provides enough pointers with some details that
Yeah.
you can find more.
Mass spec searching, I touched on that. One can search a list of precursor ions against different databases. In this case, RefMet. So you need to know whether it's positive or negative ion mode data. So let's say it's positive ion mode data. You want to select the likely ion adducts. So in this case, M plus H minus water, M plus sodium, M plus ammonium. And set your mass tolerance depending on your instrumentation. Sort by delta. So delta represents the difference between the input value and the calculated value for M over Z. Run your search. You could search from a file too. It could have thousands of different M over Z values. Check the results. Then download the results. So these are the results. So these are the input masses. So this particular input mass had a lot of hits for all of these different metabolites in RefMet. Now, some of these are known exact structures and some are not. So for example, the first one, it's 17:1 an acyl chain, but the position of the double bond is not known. The regiochemistry of the double bond is not known, so it doesn't have any structure. The second one, okay, it's a straight chain saturated molecule, so then it actually does have a structure. This is the straight chain C17, so that's why some are linked. We have some examples and some we don't have examples to exact structures. So this is obviously a big, long list of hits. All right. Anything else? No more questions? Oh, one other thing I want to point out are the tutorials. Tutorials for NMDR study submission, MW tab, the REST service. And most of the content that I've talked about are covered in all of these tutorials. An overview of the workbench, NMDR browsing, searching, analysis tools, which we just covered, MetStat, the REST service with lots of examples, the RefMet. So this is a really good place to look if you want to look in the details of what you can do with NMDR. Okay, I'll hand it back to you then, Mano.
All right. Thank you, Ryan. Thank you so much for these three extensive presentations back to back.
So, one way is to go to sample source. So, liver. That'll give you a list of all the studies. That's one way to do it. The other way is to go into MetStat and select sample source. Say, and these numbers in parentheses give the numbers of studies that have. Okay. So now if you want to see all studies on metabolites found in liver with all types of methods, that's how you do it. That's a big, long list. Well, you could filter that, obviously, based on other analytical parameters and species, obviously, and so on and so forth. That's how you do it. Also, you can select by human pathway if you're interested in a particular pathway. Say aspartate metabolism, for example. All of the metabolites involved in one particular pathway can be found using that method. So yeah, this is a very powerful feature to narrow down your search to the conditions that you're interested in, both in terms of analysis and sample source.
Well, Shashi has a question that in the search panel, can one specify multiple sample sources? It has to be done one sample source at a time.
No, you got to do it one at a time.
And the related question from Jane is, can you search for a single cell type?
That will be in sample source. So typically, you'd see cardiomyocyte cells. Depending on what you mean by cell type, if it's a different bacterial cell, then you're talking about species. So it really depends on the context. So there's a very long list of species here. Now, some studies would look at, multiple bacterial species at the same time, especially in gut metabolism. They wouldn't actually separate them out. Then other studies that would be much more controlled to be looking at one particular species. So it really depends on the study. So this is, for example, marine microbes. So we have studies on marine microbes, okay? So where the species is not being delineated, but then in other cases, you find the exact species, Pseudomonas, and so on and so forth, Staphylococcus, and so on. So it really depends.